The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, this is episode 87 of the eLife podcast. I'm Chris Smith from The Naked Scientists. And this month, can cold-blooded animals still run a fever? Yes, they can, but how do they do it? Also, equipping bats with backpacks to record the sounds of their food choices, and kingfisher colours. But first, according to various sources, about 5 billion of the world's 8 billion plus population regularly use some form of social media. Facebook alone has about 3 billion active accounts. Consequently, the societal impact of these media, and specifically the messages and the information that people convey through them, is huge. During the COVID-19 pandemic, claims that vaccines contain microchips so Bill Gates could track us were everywhere. In fact, someone even sent me a circuit diagram for the chips in the vaccine that I'd had, although when I looked a bit more closely, it was actually the circuitry for a guitar effects pedal. But did someone share and like that information because they too were amused by it, like I was, or because they genuinely believed what the post purported to say? That, UCL's Laura Globig argues, is the problem with many social media platforms. Engineered to engage and promote information exchange, they don't reward users for the veracity of what they share. So she's come up with a way to recognise and reward users for the trustworthiness of what they're sharing.
The spread of misinformation online has skyrocketed. And this has had quite drastic consequences, such as increasing polarization and resistance to climate action and vaccines. And so far, existing measures to halt the spread of misinformation online, such as, for example, flagging or reporting posts, has had only limited impact. So we wanted to know if we could help address this issue of misinformation online.
Before we come to that, with what you just said, that there's been this association between public behaviour and social media, do we know that's causal? That because of people putting inflammatory things on Twitter and Facebook and all the other places, that is translating into people not having vaccines and so on?
The causal relationship is really difficult to establish, but what we know about the spread of misinformation is that because of the advent of the internet and social media platforms, it's become incredibly easy to reach people and to share information. And that information doesn't have to be reliable. So therefore, it can also be true that false information is shared. And in times of crisis, people then turn to the internet as a source of information, and then they're more likely to trust it. And that might then lead to vaccine hesitancy, for example.
And is it true what people hear about these various platforms, that they are craftily coded so that they're almost addictive and they appeal to people in a very specific way and encourage them to, a bit like a mainline drug, take more of them?
It is true that these platforms tend to rely on metrics of engagement, so reward and punishment dynamics, to motivate people to post information online and also to react to posts. And that is very similar to any sort of reward mechanism you would see in the real world. So just like giving you money, your brain behaves the same way if you receive likes on social media. You process it in a similar manner.
And are there any particular groups who are more susceptible to this, or is everyone potentially a sucker for it?
Everyone is susceptible to a certain extent. It is true that people who tend to not question information as much, so who have less critical thinking ability, tend to fall victim more.
So what have you done here, and would it work for those people?
People are actually quite good at distinguishing true from false information. So it's not a lack of ability. In fact, existing research shows that lay people are just as good as professional fact checkers at telling apart true from false information. Instead, one reason for the spread of misinformation online is the lack of incentives on social media platforms to share true information and avoid sharing false information. People tend to choose actions that lead to rewards or positive feedback and avoid those that lead to punishment. And on social media platforms, these rewards and punishments come in the form of likes and dislikes. But the issue with these likes and dislikes is that they aren't representative of the accuracy of the information you're sharing. For example, you could like an obviously false post because you think it's amusing. So we now propose that the key to reducing the spread of misinformation online is not to tell people what's true and what's false, but instead to directly incentivize them to share more true relative to false information. And so we need an incentive structure where these social rewards and punishments are directly contingent on the accuracy of the information.
So what you're saying is, instead of there being thumb up, thumb down, like, dislike, I could have, I trust this, I don't trust this.
Exactly. So in this study, we do this by slightly altering the engagement options offered to users. So we're not taking away the like and the dislike button, but instead we added an option to react to posts using, just as you said, the trust and distrust buttons.
You can envisage why people would be incentivized to use that, because it's an additional badge of honor for them, saying, oh, I'm sharing this, but it's a bit iffy. And then if it turns out that it is a bit iffy, they can say, well, I told you so. So it does kind of play into the same reward system, but it's for the benefit of more clear communication.
Exactly. Here, there's no ambiguity in the use of the trust and distrust. So trust, by definition, is related to reliability. It's a firm belief in the truth and reliability of something. And so what we found in this study is that people would use these buttons to actually differentiate between true and false posts.
So what data have you got that suggests this will actually work?
What we did was we created simulated social media platforms. And in these platforms, users saw both true and false information. And then we added an option to react to posts using a trust and a distrust button, in addition to the usual like and dislike buttons. And so then what we found was that people use these buttons to differentiate true from false information more so than they use the like and dislike buttons. And as a result of that, to receive more trust rewards and fewer distrust punishments, other participants were then also more likely to share true information relative to false information. So what we saw was a large reduction in the amount of misinformation being spread.
Does the person effectively score points for trusting something that turns out to be true? Is that how it feeds back and endorses that, so that person's building reputation? Is that one of the incentives?
So the incentive is receiving the trust themselves. So we had 3 experiments, and in the first experiment, we gave participants the option to react to posts using a trust, distrust, like, and dislike button. And the incentive here is just to engage with the post itself. And so what we found was people used the trust and distrust buttons. And then in the second and third experiment, we looked at how receiving trust and distrust feedback from other participants would impact sharing. So there, people are motivated to share true posts so that they receive a large number of trusts and very few distrusts.
And of course, your timing is perfect, because in the UK, at least, the online safety bill is making its way through the government process. This is the idea of trying to make the internet a safer place where misinformation propagates more slowly. So really, the whole world, the business world, should be receptive to ways that they can improve, not just the engagement, and but the quality of the engagement.
Exactly. That's also our hope. And what we're doing here is we're not reliant on any fact checkers or anyone definitively determining whether something is true or not, but instead we're putting the onus on the user, which actually increases user autonomy, which also again would be very appealing to the platforms and hopefully to social media users themselves.
Well, let's hope she's right. Laura Globig there. A common consequence of infection is fever, at least for warm-blooded creatures like us. In response to various inflammatory signals, the brain's hypothalamus turns up the thermostat and our temperature climbs with inevitable consequences. We, in response, reach for the packet of paracetamol and bring it down again. But are we, by doing that, actually prolonging our illness? The fact that so many animals have this fever response to infection argues that it must serve some kind of beneficial purpose, and there is evidence to suggest that some microorganisms don't do so well when body temperature climbs. But studying this is very difficult, because if we artificially alter a warm-blooded animal's body temperature, the metabolic stress that we inflict clouds our ability to ask questions about the impact on the infection. So the genius solution that the University of Alberta's Daniel Barida has hit upon has been to work with a cold-blooded species. Because, yes, fish run a fever when they're infected too, but not by boosting their metabolic rates. They do it by moving to warmer patches of water to force up their temperature. And this means they're perfect for studying the impact of fever on immune function.
The beautiful thing about this fish is that it's very comfortable at temperatures between 1.4 degrees and 34 degrees. Goldfish, like other cold-blooded vertebrates, do not have the capacity like we do to change body temperature through metabolic means. So instead, they actually move to a warmer or colder temperature. So fish will go to a warm side of the pond and be able to induce or stimulate that fever through behavior rather than through metabolic means.
You're telling me that goldfish run a temperature when they get sick, like we do?
They do. So if you look at the pathways that are involved, they are shared. Yes, there are physiological differences, and we expect that there's going to be some differences between the febrile responses. But for now, they're largely shared. And the trigger is just behavior when you're cold-blooded versus metabolic when you're warm-blooded.
So what did you do to these fish then to make them do this so you could investigate it?
Well, that was the most beautiful thing, I think, out of this study. The fact that we were able to stimulate them with a pathogen or with a vaccine, and we simply observed them. So it's not that we really manipulate anything in the animal themselves. What we did have to do was create an environment where they would be able to actually show what that natural behavior was. And that was the most challenging thing.
What, you mean a tank that's got hot spots and cold spots?
Exactly. So this was one of the fanciest tanks that you will ever see to hold a fish. Essentially, we had a tank that had zero physical barriers, but there were multiple different temperatures within this tank. So everything was controlled through fluid dynamics. We had over 5 or 6 engineers working on this for a couple of years and basically got it to the point where we have a very discrete gradient, so different temperatures that were very consistent for over 2 weeks. And we were able to just simply put the fish there after they were challenged, after they were vaccinated, and look at what happened.
And what does happen?
When the fish encounter a pathogen, they will move to the warm side of the tank. And what was really interesting to us is now that we had that extra resolution to observe this, was that they do so very dynamically, and they will go to the warm side of the pond, and then they will stay there for the duration of their fever, and they'll come back.
If you can control the temperature in that way, you can presumably do experiments where you infect or challenge a fish and then you deprive it of access to such a high temperature. And it has to therefore, it can still choose to go somewhere warmer, but it might not be as warm. What happens if you do that?
Well, in our case, we were able to have what we call a control group that allowed us to see exactly that. And when the fish were challenged, so they encountered a pathogen, but the tank was held at a specific temperature, in this case, the base temperature that they had been acclimated to, they did not get all the benefits that we found with the tank with all the temperatures. So we didn't see their capacity to clear the pathogen being as fast. They weren't as good at controlling inflammation, and they weren't as good at repairing the tissue that had been damaged by this infection.
Some bacteria, though, grow better at higher temperatures. So is it still beneficial to the fish to go to that higher temperature, or is there a middle ground? And do they know what that middle ground is?
That is an excellent question, and that was one of the concerns going in. Fever is typically thought about as moving pathogens away from the temperature that they prefer, but yet we understand that many of these pathogens do better at the warmer temperatures. In our case, the Aeromonas bacteria that we used grew better at the higher temperatures, yet the fish were much better equipped to combat this pathogen when they got the chance to move to these warmer temperatures.
Did you look inside the fish to see what effect the higher temperature was having on their immune system and on their physiology to effect this improved recovery at higher temperature?
Yeah, absolutely. We looked at the capacity of the white blood cells to come to the site of infection, and that was much more efficient. And what was interesting is it wasn't more white blood cells coming to the site. It was just they were coming earlier. And you can imagine if white blood cells come earlier, they find less pathogens at the site because the pathogens have had less chance to grow. So therefore, it becomes a much better scenario that is in the favor of the host.
Do we know how the white blood cells improve their efficiency at these higher temperatures, what the mechanism of that is?
What we found was that at the site of infection, there was earlier production of various molecules that call on those white blood cells. The other thing that we saw was there was much better engagement of the central nervous system, and that is actually able to create what we call a systemic response. So basically, a whole body response to better equip this animal to be able to combat this infection.
And you think that the same thing is almost certainly going on in us when we run a fever. We've got the same molecules, the same cell behaviour, and therefore the same gains through running a temperature to fight infection, just like these fish are.
Well, we've been studying comparative systems, so basically different types of animals for many years. And what we find is there's a lot of shared responses, particularly in what we call the innate arm, which are those very well conserved early responses from the immune system. The second aspect is that there's well documented survival advantage, not just in fish and other cold blooded vertebrates, but also mammals. Rabbits infected with rinderpest, for example, survive much better if you allow them to have that fever response. And there's many other examples in the literature.
The strange thing, though, is that because we feel so awful when we run a fever, we end up popping pills like paracetamol to bring our temperature down. Are we therefore making ourselves potentially suffer less but for longer by doing that? Should we just grin and bear it and run a fever then?
I think there's a few factors to consider. We do think of fever as a symptom, and I think that has to change. The second aspect that is also very important is not just taking the antipyretics at the time of, but there's many people in the general public that may take this preemptively, whether it is before a kid goes up for vaccination and you simply do not want him to get cranky after, or if you are a sports person, you may think that you don't have very good ankles and you take one of these drugs right before a game just simply to prevent that inflammation from happening. Yes, you may be alleviating some of the symptoms, but the question is, are you losing something? Of course, we have to confirm this in mammals, but based on everything that we've been able to observe, it seems that at least a good part of it will be shared.
Absolutely fascinating. From the man who is officially the owner of the world's fanciest fish tank, Daniel Breeder there. This is the eLife podcast with me, Chris Smith. Still to come, we'll hear how spiders evolve their venoms and the factors that drive the development of feather patterns. But first, when I step outside at dusk near my home, I'm delighted by the sight of local bats wheeling around above my head as they emerge to feed. But what is a bat's ideal meal, and how do they balance the different sources of food, particularly as environments come under pressure from climate change or human encroachment? Laura Stilscholt, who's at the Leibniz Institute for Zoo and Wildlife Research, has been studying one species of European bat by equipping them with backpacks that log where they go and what sounds they produce as they pursue and then tuck into different food choices.
Bat populations across the globe are in decline, mainly due to human activities. So we found it quite urgent to try to understand how bats maintain their energy balance when they are hunting in the wild in a complex environment.
What sorts of bats were you looking at? Because obviously that's a big family of mammals, isn't it, the bats?
Yeah, bats comprise 20% of all mammalian species, and they live in all sorts of ecosystems, from rainforests to deserts. We were mainly interested in one of the biggest bats of Europe that's called the greater mouse-eared bats. And they live in big parts of Europe, and they eat insects at night.
Is it purely just prey on the wing thereafter, or can they tap into other sources of food?
Many bats are actually eating nectar or fruits. Some are eating vertebrates or frogs and lizards. And some bats are also eating insects that are walking around on the ground. So they can catch them a little bit like an owl will catch a mice, for example. So they're extremely versatile. And we think it's because they have powered flight and they have echolocation. And those two things together, we think, has made them develop into many different niches.
Of course, the price of that ability to fly is that they have to get a lot of food because they're burning through a lot of energy.
Yes, exactly. Because bats are small, they have a high metabolic rate in general, but because they're also flying, they have an extra added cost of transportation.
So how did you try and find out more about what they were eating, where they were eating it and how?
We tried to study what they were eating and how they were maintaining their energy balance by attaching small miniature backpacks to the back of them. And these backpacks, they have a microphone that can record their sonar. And it also has some sensors that records the movement of the bat in 3D space as well as GPS.
So how does that tell you what they're eating then?
We can then use the microphone to listen through the night of the bat hunting. So we can listen when they are capturing prey, when they munch their prey after they caught it. So we can actually try to understand their natural behavior through the sounds that they emit and the sounds that's in their environment.
And what were you doing, catching bats and then equipping them with these backpacks?
Yeah, so we went to a very beautiful cave in Bulgaria where there is a big maternity colony of these greater mouse-eared bats. So we catch them when they fly out to hunt at night. Then we put this little backpack on the back with some glue, and then we release the bat, and then it takes off into the night. The difficult part is not catching the bat, but actually getting the same bat back a few days later. So we catch it with different strategies. Sometimes we can find the tag because it has detached from the bat and fell below the colony. Sometimes we can catch it with hand nets, and other times we put up these big traps where we catch it, and we wait for many hours until the bat is flying out, and then we try to catch it.
Story of a scientist's life, especially one that works with animals. So you equipped a big group of bats with these backpacks. You let them go and hunt and forage. You then hopefully get a reasonable number of the backpacks back and get the data off, and then you build that kind of picture through interpreting the sounds of what they were doing.
Yes, based on the sounds, we can visualize how they are sensing the world, and we can then make a visualization of how they see the world, and then look through that visualization and see when are they looking for a prey, when are they flying close to vegetation, when do they land on the ground and try to catch a beetle, for example.
That sounds like an enormous amount of work, though. How do you analyse what must be hours of audio footage per animal?
When we get the tag back, usually I listen through those hours myself to just get familiar with how it sounds to be the bat out there. And then afterwards, we can then train a computer to find different patterns that we're interested in, for example. When are they catching prey in air? When do they catch it on the ground? And that can help us then quantify how much food are they actually getting in a night.
And how do they hunt? Do they go chiefly for flying insects, chiefly for what's on the ground? Are they led by abundance in those relative places and they just go where the food is? What sort of picture's emerging?
We found out that the bat seems to prefer to capture very big beetles on the ground, even though they're really, really difficult for them to catch. They fail every 2 out of 3 times they try to take a beetle from the ground, but they still do it all the time. So we think it's because they are out for this very chunky, big meal on the ground.
It's worth going the extra mile and failing a few times because the caloric reward from one big chunky beetle, that's a lot of mosquitoes you've got to eat to make up for one beetle.
Exactly. So they can afford to fail quite a bit because when they get the prey, they get a big amount of food and a big amount of calories.
So having learned this now, that bats have this particular feeding behaviour, they prioritise their feeding behaviour, big chunky ground-dwelling species if they can get it. How does this feed into how we should conserve them better?
It feeds into how we should conserve them in 2 different ways. One is to understand how they switch their strategy between different types of prey. We can see that when the environments change, they tend to switch strategy, and then they go for the more easy to catch prey, but not very caloric, and those are the flying insects. And that's a good strategy to be sort of versatile in your hunting strategies, but it can also be a problem if the environments change in a way where they are all forced to switch strategy to the less caloric efficient prey in the air.
What are the implications of that then? Because one of the issues is that human population is going up, human encroachment is going up, and human infrastructure as a part of that encroachment is going up. So what are the implications in terms of what we do that we should do differently now we know about this, or we have these new insights into the bat feeding behaviour?
When we know the feeding intake of a wild bat species like this, that can help us to estimate the resources that's needed to sustain healthy populations in an area, for example. So in that case, it can help us to inform protection strategies, or it can also help us to try to predict the responses that bats have to human encroachments in the future.
Bat lady Laura Stilshot, possibly one of the most patient animal researchers I've ever come across. Now, fans of Rudyard Kipling's Just So stories will recall the yarn about how the leopard got his spots. But now we're going to hear how the kingfisher came by his colour patterns. From the Field Museum of Natural History, Illinois, Chad Eliason.
When I look out my window and I see all these colorful birds, some birds are less colorful, some are more. I really want to understand what makes some groups of birds more colorful than others.
Now, if we asked Charles Darwin this question, he would just roll his eyes and say, it's evolution by natural selection, wouldn't he?
Yeah. That is true that natural and sexual selection are important in explaining some of these patterns that we might see, but at the same point, I think it's really important to think about not just extrinsic or external factors, but also internal factors like an organism's physiological or genetic makeup that allows a bird to produce a different kind of color pattern.
So tell us about what the project involved, what you studied, and how you went about it.
Yeah, so we studied whether the way that these colors are produced on a bird's body might be reflected at or kind of limit their ability to kind of evolve and diversify across the world. So these kingfishers are found globally, and we used museum specimens, and then we used reflectance spectrophotometry, which is just kind of a fancy way of determining what wavelengths of light are reflected from a feather, and that can tell us details about whether it's a pigment or a structure producing that color, and it also gives us a good data set to be able to ask our evolutionary questions.
And what were those questions? Because I'm envisaging you have enormous numbers of feathers from different parts of a bird's body, and you can interrogate them to ask what colours are there and what sorts of colours are there. But what do you do with that data? How do you then build that into an argument?
Yeah, great question. So there were kind of two big questions that we wanted to get at here. So first of all, I mentioned this global distribution of kingfishers. What I didn't mention is that a lot of their distribution is focused on islands, and islands are really a great way to kind of look at how an evolutionary experiment may be played out. So they're kind of good natural laboratories for studying evolution in action, because these species that get to islands are isolated. There's differences in what kind of predators are after them. There's differences in what species are around. And it's a good way to look at how living on an island might affect how quickly you evolve color. So that's one of our questions that we asked. And then also, since we had color measured across the body, 22 different spots on the body for a total of, I think, 3,000 measurements we took, we could ask whether simple and complex color patterns differ in their characteristic rates of evolution. So whether they influence the broad trends that we see in color diversity across the globe in these kingfishers.
And what did you see?
Our prediction was that we expected birds on islands would evolve color faster. And we did find that when we compared them to kind of mainland kingfishers. And then we also took this data, kind of the complexity, this plumage complexity across the body. And then we asked whether complexity affects evolutionary rates. And what we found there was, yes, it does. So if we look at birds with more color patches, so they're able to produce different colors across their body, that has evolutionary implications as a group of species that are closely related kind of spread out and evolve over time. Since they have more ways to produce color across their body, they're actually overall accruing more color diversity over time than, say, a group of closely related species with very simplistic plumage patterns and few color patches.
Why do you think both of those things happen? What's the mechanism behind both of those statements you just made?
Well, I think the mechanism for the first one, for the island pattern, is, as I was talking about, these kind of natural laboratories, but islands are, if we envision 10 different populations or species of birds on 10 different islands compared to 10 species that are interacting a lot on a mainland, there's just more potential for the genetic trajectories or the pedigrees to kind of spread out on these islands, because the gene pool is more distinct in these different islands compared to a mainland. So that's kind of one of the reasons, just this isolation, geographic isolation, allows kind of color to evolve on its own trajectory. For the other argument, I think what may be happening is that developmental maybe constraints as these birds develop There could be kind of genetic or developmental constraints limiting how color can be placed on the body or what color maybe the head is compared to the tail. And then that, I think, is what we're seeing so when we look at an organismal level and we scale it up to kind of this macroevolutionary scale, we kind of see this relationship that a more constrained plumage configuration causes just less color diversity over time.
So now we know. That was Chad Ellison. Two of the best-known venomous animal groups on Earth are the snakes and the spiders. Both produce venoms that comprise cocktails of different chemicals. Snakes do it by using an assemblage of genes that they appear to have co-opted into venom production over evolutionary time. And as a result, these genes produce venom proteins that tend to vary in the way they look and work. Spiders, on the other hand, appear to have done things differently. They've repeatedly used the same genetic template and varied it, as Kartik Sunaga explains.
So we know that all snakes in the world and a group of lizards, venom originated in the common ancestor of these two groups of organisms. And we kind of understand where these toxins come from. They're all coded by completely different genes. But if you look at spider venoms, they're all equally complex and diverse, but we absolutely have no clue as to their evolutionary origin.
So I guess the question then is, well, do they follow the same sort of path and trajectory as the snakes, or do they do things differently? And until we look, we're not going to know.
Absolutely. So that's what prompted us to undertake this study. So we thought initially that probably all these different toxins that have very different functions, they probably came from different genes, with independent evolutionary histories. But when we started looking into spider venoms, it turned out that most of them comes from just one scaffold or a template, which seems to be modified into all these different forms. So it's a completely contrasting strategy that you observe in spiders in relation to the venoms of most other animals.
I suppose we should also explain why all these animals that are venomous have a lethal cocktail. Why don't they just use one chemical?
If you're completely reliant on one type of toxin, then the prey animal can develop resistance to that toxin. So that's why all these venomous animals have very different types of toxins so that they can overcome this thing called evolutionary arms race. And another reason why you would want to hit multiple targets in a single animal is because you can incapacitate that animal so much more quickly instead of trying to just hit one target. That takes a lot more time.
So how do you think then, just sticking with snakes for a second, before we contrast with spiders, how did they arrive at the combination or the cocktail of chemicals they have got?
So snake venoms are very complex, but in terms of the type of molecules that they contain, but if you actually look at their evolutionary histories, you'll see that most of them come from some 6 to 12 different genes. So the way they have achieved this tremendous diversity is by incorporating these different genes into their venom glands, with which they produce all these different toxin proteins.
And the spiders, in contrast?
So based on our findings, we now see that all the spiders in the world have evolved this template around 375 million years ago in the common ancestors of two groups of spiders. One is called Araneomorphae, which are these orb-weaving spiders that you see in forests, whereas the other one is called a Mygillomorphae. These are big, black, hairy spiders that you see in pet stores. So it seems that the common ancestor of both these spiders evolved a template which seems to be then recurrently modified into all these different proteins or toxins that we see in their venoms today.
So the snakes basically just mutated some genes and have mixed and matched between them over time, whereas the spiders have stuck to the same tried and tested formula, but they've tweaked it.
Absolutely. So snakes have relied on recruitment, meaning that they would recruit completely different toxin encoding genes into the venoms, whereas spiders have just used one template and extensively modified that or innovated that template into different toxin forms with completely different functions and activities.
And how do you pin this number on 370 plus million years ago as the origin of this particular strategy? How did you arrive at that date?
So what we did is we collected sequences of these toxins, these compact toxins called disulfide-rich peptides. In short, these are very small toxins which are abundant in venoms of the majority of spiders around the world. So we collected all these sequences from these spiders, and then we looked at their evolutionary histories, tried to figure out where do they stem from. And by doing that, we figured out that we can trace it to 375 million years in the common ancestor of these two groups of spiders that I mentioned earlier. And we call it Adi Shakti, or the creator of the original universe according to Hindu mythology. And we propose that this template is what was used by these spiders to generate all the extent diversity that you see in these compact toxins in spiders around the world.
One therefore has to wonder why at that point in history they hit on that solution and stuck with it. Was it just that it came along and it was very successful, or did they get it and repurpose that strategy from somewhere else?
Unfortunately, we don't know the answer to that question. For example, in snakes, we do see that certain lineages have evolved certain types of toxins the moment they evolved this high pressure front fang for delivering venom. For example, if you look at cobras and kraits and sea snakes, you see that they evolved this high pressure front fang venom delivery system around 54 million years ago. And we see a parallel diversification of one of the most important toxins that we see in snakes called three-finger toxins. So there we have a perfect correlation between the evolution of the venom delivery system and the diversification of the toxin. But in spiders, we don't know.
Kartik Sunagar, he's at the Indian Institute of Science, Bangalore. And that's where we have to leave it for this month. You've been listening to the eLife podcast that's produced by The Naked Scientists. There are previous editions, the references, and also the full text transcripts for these programmes, as well as details on how you can subscribe to this podcast at nakedscientist.com/eLife. The Naked Scientists also publish a weekly science programme which covers the latest leading science stories. You can find that at nakedscientist.com/podcast. I'll be back next month with another look inside eLife. Until then, though, from me, Chris Smith, thank you for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.